Okay, good afternoon, everybody. Thanks for joining this session and being on time. We will start now, and probably, if we're lucky, then also the fifth presenter will show up and show us what he has to show us. So this session is on technological innovations for implementing the Sustainable Development Goals. And I think we have quite a nice mix of different perspectives. So as the SDGs themselves are multidisciplinary and have a lot of a very broad, a very broad spectrum of topics and challenges. We also have quite a broad spectrum of presenters today, both geographically and content-wise. So we come all the way from European applications to Central Asia, India, Sub-Saharan Africa. So we have really a broad thing, and also topic-wise, it's from starting from cosmetics over water management, gender perspectives in agricultural production, and so on and so forth. We will see the fourth presentation might not take place, and if that's happening, then we have a bit more room for discussion after the presentations. Otherwise, we have around 10 minutes for the presentations and 5 minutes question and answers directly after the presentation. And then in the end, we have a bit of room to have a wrap up and a broader discussion. Okay, so then I think we can just start with the first presentation from Binal Doktoria from Rheinwahl University in Germany. Welcome. Thank you for coming here. And
So, very good afternoon, everyone. Thank you for being present here. Myself, Binal Dobaria. Just a brief introduction. By profession, I'm a microbiologist, and I'm working on this project for Super Chain project. And so I would like to first thank you, Boku University Vienna, for organizing this conference on international research on food security, natural resource management, and rural development. I'm mentioning the topic of the conference because our research is also aligning with this topic, which is utilization of nut and fruit processing byproducts for sustainable and cosmetic formulation. It is a pathway for microplastic-free beauty products. So I would like to start with the little journey, which starts from the Central Asian country, Kyrgyzstan, which is a large agroforestry, which is having a large agroforestry site for walnut trees, and it is producing 1,000 to 3,000 tons each year walnuts from agroforestry sites. Additionally, it is also having a good agriculture sites for the apricot trees, which producing 16 to 17,000 tons annually apricots from them. And they are having the good facilities for the fruit drying processing line, which is good for the drying of the fruits and exporting the fruits to the different countries like Russia, Turkey, and more. But the problem is that they having a less optimized fruit processing line. So the waste is more than the beneficial part from the fruit processing line. So our goal is to utilize these waste streams into a useful way that it could be benefits to the farmers of the Kyrgyzstan and also to some company in Germany. Right now, they are upcycling the ingredients like nuts from the apricots, for example, to extract the oil from the apricot nuts, and utilizing that for the cosmetic products and other as a serum product development. And the end product after extracting the oil from the nuts is being utilized for the feed product for the animals. But my focus line is on the shell, which is, I think it's too good to be waste, because imagine if you are producing if the trees are giving the 10 kilos of fruits, and after processing 3 to 4 kilos of shells, which only goes to waste. And yeah, it's not good idea to just waste it or just burn it for the energy production. On the other side, as you all know, the microplastic waste in the marine and environments, it's growing and growing. So for the last year on 25th September, European Commission has intentionally added microplastic products to the cosmetics and detergents and medicines and many other products, which has no particular meaning for adding the microplastic into the product and then end up into the waste, which is not recyclable. And it is good part that it will prevent half a million tons of microplastic going into environment by this just a small change. And on the other side, it is a good opportunity to introduce the alternative in cosmetics. So the idea is to use the walnut shells and apricot kernel shells as a natural biodegradable alternative to the abrasive particles in the cosmetics, and it will add the economic value to the farmers in Kyrgyzstan, and in worldwide it will reduce the microplastic waste. So these are the objectives of this research project. So basically, it will start with growing the good quality of walnuts and apricots, and just to have a good quality of raw materials for the cosmetic product development as the walnut shells and the apricot shells. So as you can see here, the walnuts and apricot shells have a very sharp edge, so it's difficult to get the smooth edge powder from that. So the first challenge is to overcome this challenge and having the 50 micrometer to 100 micrometer smooth size edge powder. And next is to incorporate them into the cosmetic product as abrasive particles. And after that part is to use ensuring the microbial stability of this product. And the last would be the pilot scale processing line development. For this, the methodological part starts with the sample collection and preparation. I'm especially mentioning the sample collection and preparation part because it involves 500 grams to 500 kilos of the sample collection, and not only collection and transfer of this huge amount of the shells from Kyrgyzstan to Germany. That involves lot of efforts and documentation process, thanks to our coordinators who are maintaining these things very efficiently. And the next step will be the grinding process partners in Germany. They will optimize this grinding process, and then my part will come to ensure the microbial analysis, which will ensure the quality of raw material and pre-product and the end product. And the developed formulation will be checked for the adaptive preservation test based on microbiological analysis. So next, I would like to explain a little bit of the microbiological results from my side, which is you can see here in two graphs. So it shows that it has effect of harvesting time on raw materials. So what I did is I checked for the bacterial and fungal community on the shells during the different time intervals. So first sample was collected in 2022. That was after 6 to 8 months of the storage time, and the batch 2 in 2023 was collected just after harvesting the fruit and processing of the drying process. So it shows that the stored samples have less bacterial and fungal community in walnut shells compared to the freshly harvested shells. And the same, if we check for the apricot kernel shells, compared to the walnuts, the apricot kernels having more bacterial fungal community because they also having some pulp fruit part on the shells, and that is more prone to the microbial contamination, and also it increase into the freshly harvested sample. Next was like we have our idea on the bacterial and fungal community, and then now we wanted to know like which kind of community on the presence on the shells, and that was done by the metagenome analysis, and the result shows here in the heat map, which shows that like 7 to 8 kind of different cell sample was analyzed for this, and it shows that so in apricot kernel shells, majority it's a fungal community, which is Rhizopus, Saccharomyces, Plasmabara, and some bacterial community like Klebsiella and Halomonas. And it was important to know to the source of contamination, which shows that this community is majority coming from saline environment, soil environment, plant materials, human affecting things like skin and human contamination others, and the fermented food. Same for the walnut shell shows that a higher abundance of the bacterial community like Abbdibacterium, Cutibacterium, Methanobrevibacter, and more. And interesting to know, it is also coming from marine environment, human skin, animal, desert soil, some thermophilic conditions, and the plant material waste. And then after the metagenome analysis, next step is to use this bacterial community for the adaptation and preservation test, because right now we are doing the preservation test according to ISO 11930, which is involving the bacterial community like Pseudomonas, Staphylococcus, and Escherichia coli, and the fungal community for the Candida albicans and Aspergillus brasiliensis. And we formulated 3 base formulation for this project, and which is all in water formulation, water in oil formulation, and the rich cream. This is right now, it is without the raw material inside. So just to check the preservation efficacy of this base material, we check with normal preservation test protocol, and then after we will check it for the abundance community according to the metagenome analysis, as I showed in the previous graph. So by this research, we are hoping for the no poverty on the zone, because we are also helping the farmers to have a circular bioeconomical benefits from the fruit processing line, and the beneficial to the industrial innovation by reducing the microplastic. And another way, it is a responsible consumption and production line from the both way. So this project is not ended yet. So next steps for this future research direction, which involves the storage condition finalization for raw materials, because we have to finalize the temperature, humidity, and other affecting factors for the raw materials, and to have a good quality and constant quality of the raw material, and ensuring the economical and environmental benefits also will be achievable by this project, and ensuring the product safety and quality management will be achievable by this, and the last is to making the consumer awareness, which is most important, because now people are not much more aware to use the biocosmetic product compared to the normal one. So yeah, this is the future research direction for this, and with this I would like to thank you, my professor, supervisor, and my working group, Hygiene and Microbiology, Sufa Chain Project team, and the coordinator of the Sufa Chain Project team, and Hochschule Rheinwald, and many people who are contributing this project from Kyrgyzstan and Central Asian countries. If you have any questions, just try to contact me or have more look on the Sufa Chain website. Thank you so much for your time and being present here.
Yeah, thank you for the presentation, for trying to be in time. You had a lot to present. Yeah, I think we have like 5 minutes for questions. If there are any, there are already, there's one. There is someone else, or just one so far? Okay, we'll start with you then.
Yeah, thank you very much. And apologies, my question is really an understanding question because I'm not a user of beauty products. Maybe I should in the future. I don't know. It comes with age. What is the microplastic for? Why is microplastic being used in these beauty products? What is it for?
So it is intentionally added into the microplastic, just like abrasive material. So for just having a texture, to having the peeling effect on the skin. So that's not one kind of microplastic. There is a mixture of microplastic which was added into the products. Sometimes so it has not any effect on the skin, but it's just a mechanical thing on the cosmetics.
Yeah, do you also have a question?
Thank you for the nice presentation. My question is regarding, does this product or this material has to go through authorizing regulations, like through clinical trials to be applied or to be commercial available in the end? Is this something that's also considered in your research or projects?
Fantastic. So I can mention that topic, but we are connected with the cosmetic company, which is also in Germany based, and they are developing this formulation. And then after checking all the microbial testing and other testing, it will be in the market available.
Thank you. And is it easier because it's a natural product, or is it so difficult to get through? Okay, so it's easier to get the permission. Okay, thank you.
Are there any other direct questions? Yes, we have like one short question, and then in the end we'll have one answer.
Yeah, just thank you. It's just for my understanding. So you are mixing two shells, I mean, from the apricot and from the walnuts, or how do you understand this?
No.
Because in your first picture, you just show some, arrow that's going from here. Are you adding them, or you treat them separately? You have a product that's coming from the apricot and the another one from the walnuts. Okay.
As I showed in the last slide, there was base formulation, and then now we will add separately the apricot powder and the apricot kernel powder and the walnut shell powder into each, and then we will check for the microbial community of the advice, like which will be stayed, that will be selected for the final product.
Okay, thank you.
Okay, thanks. Okay, then I think we will move to the, thank you for all. It was very nice. And now we're going to move to the second presentation. Is it, yeah. That's Anne-Catherine Rosenbaum from University of Sevilla, Spain. Huh?
I'll try to not yell at you. Okay, hello everyone. Thank you very much for attending my presentation. I'm very, very excited to talk to all of you, especially because We all come from so many different backgrounds and we all come from so many different scientific perspectives. But there's one thing that sort of touches all of our research, and that is water. None of our research would be possible without water. And today I'm going to talk about water to you a little bit, and I'm going to talk about what challenges we're facing in case of water availability and allocation, and how to monitor crop water stress in irrigated rice cultivation, especially in southern Spain. So we all know the deals, we all know the numbers. Estimates by, for example, the United Nations say that within the next 20 to 30 years, we have to double our food production. And we also know that watersheds are ecosystems. We know that water provides life. We know that water serves other livelihoods and serves other ecosystems, but we also know that water is precious and water is scarce, and the availability of water is decreasing. So especially in agriculture, we have to make sure and secure that we are using this scarce research in a smart manner and be aware of the importance of water. Let me take you to southern Spain. The little dots, that is actually very funny. I've talked to a couple of people here, and the first thing that when I talk about my research, what happens is that people tell me, I didn't know that there was rice in southern Spain. And as a matter of fact, there is. We have approximately 40,000 hectares in Andalusia, and it's irrigated rice, it's lowland rice, and the water is taken from the Guadalquivir River and then distributed over a channel irrigation system to the different rice plots. The problem is that it rains less and less, and these low water volumes in the river allow the saline water of the Atlantic Ocean to push into the river and ultimately increase the salinity concentrations in the irrigation water. So we basically have two problems there. We have little water and we have the salinity. In theory, what they then do is we have water reservoirs and they release the fresh water to sort of push the salinity and push the saline plug back to the Atlantic Ocean and secure that we have low salinity concentrations. But as you can imagine, that requires a lot of water. So, especially in that zone, it's water shortages and droughts are part of what farmers have to deal with on a daily basis. And what you can see in the graph above is that the reservoirs are empty. There is no water for wasting it just to push salinity down the river again, which results in the production system there being extremely volatile and being extremely vulnerable. For example, in 2023, there were only 3% of the area in Andalusia that's designated to rice cultivation actually cultivated. It looks like you're walking on Mars because you have this high salinity, which means there are not really options yet to introduce another crop, which led us, well, so one of the ideas that they're having is switching from lowland rice to upland rice or to irrigated rice. But that requires monitoring because, yeah, rice is sensitive to drought stress. So our research question was that to see how reliable and effective infrared thermometry and spectral indices are for monitoring this crop water stress. We did our experiment on an experimental plant where it's the Center for New Water Technologies. It's close to Sevilla. And what they do there, you see these plots over there. And what they do is they're using reclaimed wastewater from the communities around, and they actually do trials in olives. It's the orchard you see in the center above. And these are our rice plots. We have 6 plots, each one of them has 100 square meters, and we had 2 treatments. We irrigated 3 crops at 100% of crop water requirements, and the other one at 90%. We seeded our fields with the local indica rice cultivar. It's called Hispalón. It's of high agronomic importance down there. And we adjusted our cropping cycle to the cropping cycle, how they actually cultivate the rice in Sevilla. The rice is irrigated over drip irrigation, what you can see there on the pictures a little bit. These are pictures from our plots from 2023. And then what we did and what we have in each of these plots is we collect climate data and we collect the canopy temperature. This is the little sensor over there. That sensor has the infrared thermometer and an NDVI sensor. And with these two, we calculated the non-water stress baselines. The non-water stress baselines, you need that to ultimately calculate the crop water stress index. It gives you basically the idea of the temperature difference in which a plant is not stressed, and then of that you can calculate how the plant reacts in the crop water stress index if the temperatures are rising or if the plant is experiencing more stress. And that is the formula. Additionally, like I said, we took the NDVI index as a reference. We collected also water volume and soil moisture data, and we used stomatal conductance as the physiological variable to measure something actually on the plant and then to validate if our index is actually working or if our methodology is actually working. Yeah, so the first graph that you can see on your left is the temperature difference in crop temperature, or between air temperature and crop temperature between the two treatments. T1 is the treatment that is irrigated at 100%, and T2 is the one that's irrigated at 90%. So what we can see already, or what is a very basic information, but it's very nice to validate what's actually going on in the field, is no, the plot with the full irrigation has lower temperatures, and the one that is irrigated with deficient irrigation has higher temperatures. And it's logical, but what happens in reverse is that the NDVI with the water stress plants is actually lower, which means they actually experience slower and less development than the fully irrigated plants. And this is the whole movie that we did. What you can see here, I'll walk you a little bit through the graph, is we have the two black lines. One is a dotted line, one is the full irrigation line. And these two lines show us the crop water stress index. Now, what's interesting or what's noteworthy is that we would assume that crop water stress, or that index is actually low or close to zero when we're irrigation at the full crop water requirement. No, but we have, does this have a laser pointer? Yeah. Good. So no, we have these two at the beginning of the crop cycle and at the end of the crop cycle. We are higher than zero, or we're in high concentrations or levels of that index that we wouldn't have expected. And that is where the NDVI comes in, because with the NDVI, we can actually validate and see that these measurements, or why it's actually so high here, is because we still have bare soil. Like the plant is not grown enough yet to cover the soil. And that is why in the crop water stress, it appears as if it would be a stressed plant. And then in the end, it's because of the senescence of the plant. So no, the plant actually, is maturing, and that affects stomatal conductance, but the crop water stress index reads that as a plant being actually stressed. But because we know we have the NDVI and we see that the NDVI is also going down, we can, we know that it's because of the plant senescence. Oh, right, the picture. So no, what I just explained on what's happening here, you actually see that in that picture that the soil isn't actually covered yet. And up here you see that the color of the fields are actually changing due to the maturity of the plant. And this is somewhere around 60 days, I think it was. These are my pictures. They are actually from that trial. So this is what the field looks like throughout the cropping season, right? And then finally, Our measurements of some of the conductance were also lower in the drip irrigated fields and the deficient irrigated fields, and that aligns with our results. And that is actually what we wanted to do with the stomatal conductance, no? So as a conclusion, we have that mild water deficit caused reductions in vegetative development in rice water status, and that the crop water stress index has proven to be a good indicator of the crop's water status. And during phenological stages where the vegetative cover is still incomplete or is already again incomplete, the NDVI index can be useful for interpreting the crop water stress index values. These are our project partners, and our conclusions you have here as a take-home message. And if you want to get in touch with me or ask me questions, here I am. Thank you very much.
Thanks a lot. Are there any direct questions? Maybe understanding or anything else? Yeah? Yeah, you have a question?
Thank you. Thank you for your presentation. I would like to know if this index and this method could be applied to other techniques for rice cultivation. For instance, this system of rice intensification, that is SRI, that is kind of dry, and then it's humid, dry, humid, dry, humid. Then just to know if this could be also used for that.
Thank you very much for your question. The problem, the challenge that I see, my honest answer is I don't know, so I'll walk you a little bit through my thinking process, is we're doing that on bare soil right now, no? If you do NDVI calculations, for example, on irrigated fields or on lowland rice, what happens in the beginning when you do not have coverage of the water table is what happens is that the NDVI turns negative because it reflects the light. I am very sure there is a way to correct this. I cannot tell you how it works, but I'm very sure that there is a way to apply that.
Any other questions? We have time for one brief. Okay, I would have one.
Yeah.
How sensitive are these measurement instruments to fluctuations in the reused wastewater quality? Or is it always like, do you expect there's always the same quality coming in?
The quality of the wastewater does not affect what we in the end do is we're measuring light frequency also, no? And because we're doing drip irrigation, the quality of the water will not affect what the thermometer is actually measuring. We're always at 45 degrees.
Yeah, okay, good. Thanks, then we can move to the next presentation. Thanks a lot. Yeah.
Hello, everyone. Thank you for attending my presentation. I am Dipali. I work with International Rice Research Institute, and I'm based in India office. So the presentation the paper that I'm going to present today is a part of CGIAR initiative on gender equality. And I'm currently involved in work package 2 of CGIAR initiative on gender equality, where in this paper, we have tried to explore how climate smart technologies work for women farmers based on the insights that we have gathered from the socio-technical innovation bundling approach across various cases that have been studied in India. So talking about the rationale behind the study, we know that there has been substantial evidence that technical and technological innovations, in the agri-food system, they have increased farmer resilience. But these interventions, they are found to be quite exclusive in terms of the improvement that they provide in the well-being, being women and vulnerable people being disadvantaged. While it is important to have the technological interventions, we know that alone they cannot achieve, they cannot make these agri-food systems sustainable and inclusive over time. And that's why in our initiative as well, we currently focus on combining technical, social, and technological innovations, which together we call as socio-technical innovation bundling. So as I mentioned, like we know that there are a number of interventions available, but still the uptake is low, especially by the women and the vulnerable communities. There is a recognition for socio-technical innovation bundles, which we call as STIPs, but there is no systematic evidence like whether these tips empower women, how to design these tips when someone is starting a project from the scratch, say, and how can someone implement context-specific, climate-smart, and gender-responsive tips. So in this initiative, we are leveraging upon existing CGIAR and non-CGIAR innovations that are currently happening and that are utilizing the best practices for bundling innovations. And we are trying to distill out these valuable insights so that we can apply them in our work. So I have been talking about these socio-technical innovation bundles, but what does this actually mean? So this is a bundle of social, technical, and technological innovations. Technology is something that is a sort of product or process that will improve or enhance the uptake of farmer, like that will improve the women's resilience towards climate change. Technical innovations is something like the technical skills or capacity building that is given to the farmers so that they have those skills to use the technology that has been created in the lab, to use that on the land. And social innovations, they refer to the reforms around institutional structures, cultural practices, and maybe policy governance. So that may be gender transformative approaches, social and behavioral change, and policy adaptation. It is important to have the bundle of these interventions, but it is also important to understand like these bundles are not just standalone bundles of interventions, but they need to be very systematically planned. They need to cater the needs and challenges of the local context. And also they should address the diverse needs of all the communities. That is, they should be gender responsive. Talking about the objectives, so with this background in mind, we had these objectives like documenting the cases where these bundles are, where these technology interventions are bundled, may be deliberately or inadvertently by different organizations, and examining how their perception and understanding of this technology and these technical and social aspects of this technology varies across different regions and livelihood systems in India, and generate evidence on this bundling approach as an agricultural extension system so that we can advocate for learning, scaling, and policy recommendations. So we have used case study methodology. We have used 4 case studies across India, and the data from these studies was analyzed in a qualitative manner. These are the 4 organizations. So presently, these 4 case studies, they themselves are like standalone piece of their research. But here I'll be talking about these the bundles that they have used in their approach. So these were like 4 organizations that have their different projects in different states of India. So India has a remarkable diversity, and these alone research, they present like unique context, and broadly they are showing a broader perspective of the subject. So talking about the brief about like what these organizations were aiming at. So one was working toward the promotion of biofortified crops. The other one, the second, ISST, these are the 2 names are there because these 2 organizations were together working in the state of Gujarat, where their purpose was to enhance women's empowerment and resilience through a comprehensive set of interventions, including various aspects. And then the third one, like SSCOP, which was working in West Bengal, their purpose was to focus on the gender inclusive strategies of a project, which is like SRFSI project, which was working there, and it lasted for around 5 years. So to distill out the gender inclusive strategies that have been used in this project. And the last one was SSP organization that worked in Maharashtra. They have worked on this WCRF model, women-led climate resilient farming model, and they have also focused on what are the different components of this model, how different interventions were bundled, and how they reduce the gender disparities. So the different interventions that were a part of these different studies, they have been categorized into different categories of social, technological, or technical bundles. If we talk about the technological interventions, so mainly they include all the relevant technological support that was required, tailored to the project, was given to the farmers. Per se, in case of GFI, their purpose was to promote the adoption of biofortified crops. So they provided free seeds of biofortified mustard. And then like in case of ISST, they provided they had a package of practices which advocates towards sustainable farming, and they provided the other technological assistance with that. SSCOP also provided availability of certain machineries through custom hiring centers that would reduce the burden of the financial burden of purchasing for the farmers. And the SSP provided technological support through different interventions. These all technologies were bundled with technical assistance, like providing the training, how to use these innovations, and then like comprehensive trainings were included, knowledge sharing sessions, exposure visits, all of that was included, which is mostly included in all the projects. But what stood out among these organizations in the bundling process was that the social interventions that they have included. Like most of them approached the field with the through the women collectives, like approaching the field from the women collectives helped them to tap on the community relationships that were there to enhance the adoption of these technologies. And among all of that, like ISST, Uthan, and SSP, these two organizations, they adopted a systematic way where they had women lead farmers who were having a control over a group of women farmers, and they were guiding them to use these technologies, and they also had that kind of responsibility and leadership, due to which the results were quite impressive from the field. And they gave them name like Krushi Sakhi, which is like a Hindi word which means farmer friend, or Krushi Samvad Sahayak, which means like farmer communication advisor. So they approached the field like in a very systematic manner there, and in that way they could tap on the peer-to-peer learning thing more effectively. Talking about the impact on women's empowerment and resilience, so it helped women leaders in gaining the crucial skills so that they also have the confidence to drive change and advocate for women's rights. Then they had, because of this knowledge and confidence, they could have more involvement in the decisions related to land and agriculture. And this deeper integration of technologies, what we say, into the socio-cultural and economic fabrics, have definitely enhanced women's agency. And while all of these organizations were focusing on climate smart agricultural technologies and the provision of modern technologies, so it to some extent has worked towards the resilience part and also reducing the drudgery for the farmers. Working alone has built a sense of sisterhood, and it also derives towards collective empowerment. While they also had some, like in one of the organizations, they had this structure where a person from locally from the community was working as a paralegal worker, like a community person who had some information or knowledge about how these legal rights work, and was providing a kind of socio-legal psychological support to women farmers around their land rights. And it built their identity as farmers. So while this bundling process had significant, or we can say some notable changes in the community, but there were some gaps. Like there was, in most of these studies, it has been found that there was limited involvement of men, which is we think a priority if we want to drive equitable change in the society. Then there was also a notice that they didn't pay recognition to women's unpaid care work, and sometimes these interventions added on to their existing burden because they also have domestic duties. And there was a bit of fragmented bundling of these interventions. So what we advocate in our CGIAR initiative on gender equality, where we are working on STIPs, we say that we should adopt an intersectional approach in designing these climate smart technologies. we should focus on towards making these interventions gender transformative, which means we just don't need practical solutions on the ground, but we also need these solutions to drive a shift toward to cater these societal gender norms and beliefs. And then the last one is that, as I earlier mentioned in the beginning of my slide, that it should be systematically bundling of the interventions and not just ad hoc bundling, which is required. At last, acknowledgement to all the organizations who helped in this study. Thank you.
Thank you, Dipali. It was really nice also. Are there questions? Oh, yeah. So maybe we start here and then we hand over.
Very much. Thank you very much for your presentation. Uh, at the methodology, you mentioned qualitative studies as well as case studies. So I was wondering what's the qualitative component of the research? I couldn't find it.
Thank you for your question. So basically, these different studies, they were given to different organizations who have themselves qualitatively conducted these studies in different areas. They have approached the field with some qualitative designs, that they have their standalone qualitative designs, which I haven't included in this presentation because my aim was to synthesize the results. And also, I have also, like, across these studies, we have arranged these results qualitatively, like according to their themes under different interventions. I hope that makes sense.
Okay, thank you for your presentations. Actually, I'm also working like climate smart agriculture, so actually you define it, classify social, technical, and technological innovations. I also classify it like soft and hard technological innovations, soft and hard. So I want to know that you give an example of biofortified master seeds. As far as you know, the adoption rate climate smart agriculture is still low. Also in India, in this my Bangladesh also low. So what about the status of adoptions, like the women of biofortified? Do you determine it, the adoption rate, or how it influences the livelihood of the women farmers?
So in that study, that study was a pilot study which was conducted in collaboration with the government in that area, where the seeds were provided free to the farmers. And so the adoption rate was not like a significant extent, because a few people adopted it on a pilot scale since it was provided free, but there was not continued adoption of those seeds in that.
Okay. Thank you.
Any other questions? No, maybe I also have one, because you said this should be addressed more systematically in future projects. Do you see what's the role there from a funding organization, or who should be, let's say, demanding this kind of systematic perspective or this systematic approach? Do you see that there's like research organizations who should encounter this in their research proposals already, or?
Yes, so right now, we also had a workshop session on the very first day. So this is a like side study that we conducted to generate evidence. So presently we are working in the work package 2 of, as I mentioned, CGIAR initiative on gender equality, where we are working on these tips. So we have a platform developed, which is known as, we call it like a multi-stakeholder platform. We have named it as a learning lab. So it is a pilot project, and we are going to complete the first phase of this project in this December. And we also had some remarkable results from that, where we have used this socio-technical innovation bundling in 2 distinct learning lab locations in India. And also the same work is going on in Africa, in Kenya and Ethiopia. So we have some like results, and we are advocating for its continuation. Yeah, thank you. Thank you for attending my presentation.
No, thank you. So the fourth presentation that's on digital innovation for sustainable crop livestock intensification in sub-Saharan Africa will not take place now. We'll just skip it, unfortunately, and we'll just directly go to the fifth one, and then we'll have more time afterwards to discuss.
Yeah, welcome everybody to my presentation. My name is Robin Marz. I'm from the Technical University of Darmstadt, from the working group of biomedical printing science. And my research is about the mass production, industrial mass production of cultivated meat, which is related to modern agriculture called cellular agriculture. And as you can see on the left side, I brought an example for the traditional agriculture. And from my opinion, and also from other opinions, those two fields should, in contrast of coexisting, merging, or fusing together. And this is more or less the reason why I'm here to inform you about the field of alternative proteins, cellular agriculture, and cultivated meat. And I will start with giving you an overview of what are alternative proteins actually, and then give you some examples of highlighting how traditional agriculture can contribute to this field to make it a solution to a very important problem. And then I will complete my presentation with giving an application example from my own research, which might be interesting for you. Okay, so alternative proteins is a solution to meet the global demand of proteins or of meat because the global demand rises and will be at least 50% more in 2050, according to some studies. And it's defined as meat and seafood that is generated from plants, fermentation processes, or from directly cultivating cells in the laboratory, with the idea behind it to not cycling them through animals before using them. Everything with the motivation behind it to reduce the environmental impact, GHGs, and so on. And I dared to brought some LCA data for beef from the academic and industrial research, where it's said that we can reduce the environmental impact of beef, according to the GHGs, of 90%. We have to look at those data very critically because every study is using its own methodologies. The energy sources are different and the demands for meat also, but still it could be promising that the global impact could be reduced using alternative proteins. If you look at the ecosystem of alternative proteins, so at the value chain, it all starts with the raw materials, ingredients, and inputs, and then goes to the end products. And before I come to this part, I would like to show what end products we actually have in this field. So it all starts with the most basic product, which is plant-based meat. You probably have eaten it already, like Beyond Burger, patties, and so on, which is just made out of plant-based ingredients. And then there is the hybrid meat, which is those plant-based ingredients mixed with conventional meat. I've never eaten it myself, but maybe you did. And then comes the cultivation level. So cultivated meat hybrids is you take these plants ingredients, but instead of using conventional meat and mixing with them, you use cells that were generated under laboratory conditions without using the animal. And at the end is the highest level, maybe the most difficult one. It's pure cultivated meat, so pure cell mass from the laboratory. This is the most expensive one and has the most challenges. I will not talk about it much today because I would need at least 10 more minutes. So I will concentrate later on another product type. But before, I want to show you, so the material sources, so the living cells come actually from tissue biopsies from livestock. It's like when you go to the hospital and you get like a tissue biopsy, it's nothing else for the animal. And then the cells that are important for meat, like muscle cells, fat cells, and so on, are isolated and cultivated in bioprocesses, so multiplied, and then turned into the final product. And the plant-based materials, of course, come from traditional and modern crop growing methods and technologies. And there are also already approaches, for instance, to use agriculture waste for it. So I will now focus on the next slides on cultivated meat hybrids, because it will make the first majority of products on the market. You can already buy it in Singapore, actually. I never tried it myself. And it has a great potential to lower the cost and increase the nutritional profile of the products due to the plant-based ingredients. And the data I will show you come not from cultivated wheat hybrids actually, because there are no data yet. There's like one product on the market, so no data to do any statistics and so on. So the data I show you come from plant-based meat, but we can directly transfer it to cultivated wheat hybrids. And the process of making plant-based products or cultivated meat in the end starts with the crop development, where you select crops and then you optimize them towards the protein content, because as you will see on further slides, protein is the most important ingredient that we need for such products. And then the ingredients, so the crops are fractionated into proteins, oils, and so on, and then manufactured into textured products using certain technology. I will also show some examples later on. And in the end comes the formulations to make it yummy, to make it acceptable to the consumer. So let's have a look into crop development. And this is something where traditional agriculture can mostly contribute to if we look at such product types. Because historically, all the crops were optimized for generating starch and oils, but never for proteins. But proteins is the ingredient we need for plant-based products. And as you can see, like the main crops that we use is soy, pea, and wheat. And you see like 9 of 10 products from the industry has soy ingredients. But it's just because it's abundant, you find it abundant in side stream processes of some technology processes or industrial processes. But it's necessary to expand the sources via other protein-rich beans, seeds, and grasses, because those three have their limitations. For instance, so how can you crops, or towards what can you crops developing beside the protein type and its yield? For instance, for its nutritional value, and this relies a lot on the amino acid composition, which contributes to nutrition, texture, and taste. So plant-based proteins are very different to animal-based proteins, but we want to make an animal-based product. So we have to optimize crops towards an amino acid composition that is similar to the one that we find in the animal. This is not easy, but I think you have the right methods to do that. So let's sticking to the amino acid composition, because not only the composition is important, but also the sequence that leads to the protein structure. And we're coming closer to the manufacturing process, which is also part of my research. And the protein structure impacts the protein function. And as I already said, plant and animal proteins are very different, and also in their structure. So here are 3 examples of very abundant protein structures. And the fiber structure is the one we find in meat. So all your muscles and the meat you buy is mainly made of collagen, which is fibrous, and also like the myofibrils in your muscles that allow you to contract your muscle are made out of fiber proteins. But for instance, soy protein isolate is a globular protein, so it cannot form these fibers, and then you lose the texture in the product, or you have to apply very difficult processes to get the texture. And but for the example of a soy protein isolate, you can use physical and like chemical treatments to get native soy protein isolate into a gel by aggregation. And this is interesting because such a gel or such a biomaterial is the input that you use to produce such products. As you can see, as here, one research example from me, where we printed a scaffold that is mechanically stable from soy protein isolate. So now we're coming to the processing step, and it's estimated that in 2030, plant-based meat should or will comprise roughly 6% of the global meat market, which makes a lot of million metric tons, and it has to be ensured that those metric tons can be produced. The major technologies used so far are extrudating technologies, low and high moisture extrudates, and you get such a construct from it, which compromises of fibers, so it's quite similar to your muscle, but still, as you can already see, it doesn't mimic like a whole cut of meat, as you know from the supermarket. There are consumers that still want to have their steak, for instance, like a whole cut of meat, and not only patties. So this is one limitation of this technology. Another one is it's very energy intensive and like loses nutritional quality due to its process. So I can tell you there are no technology yet that is capable of combining the reproduction of the complexity of a real steak and to be economical for large-scale manufacturing. And there's a simple solution just to explore and to investigate in new technologies that combine complexity and economical scalability. And this is where our institute is researching on. What we do is we are the Institute for Printing Science and Technology, and we use traditional printing technologies, offset printing, and so on, and try to turn them into bioprinting technologies. And in my project, which was like the first project on this focus, I developed a 3D bioprinting technology based on screen printing, which like everybody of you wearing a t-shirt was probably printed using screen printing. It was not that easy to make it three-dimensionally and to use biomaterials and to print scaffolds. But in the end, we kind of managed it. It all starts with a template. So what I actually want to print, I want to print a steak that is kind of textured. And then we developed this biomaterial and three-dimensionally printed a scaffold that is also cell-compatible, as I will show later on. We kind of got an advanced texture using also a soy-based fat material. And by coloring, it's getting closer and closer to a real steak. And it showed, I didn't include the video where I have it in a pan and like turn it around, unfortunately, because I thought the time is not enough. You can still see that it's not as close as it should be to a real steak, and it's only a prototype, so it was not our intention to make a product, but developing a technology that allows it. And last but not least, this is how it looks like when you cultivate muscle cells on such a scaffold, because we want to produce hybrid products, because if you eat meat, there's also the nutritional profile of the cells. And we also need to get this. So this is like a microscopic image here of these little cavities, hexagons, and those are stained muscle cells. So the green ones are just elongated muscle cells, and the red ones already express these proteins that allow you to do the contracting. Yeah, this is the end of my presentation. The take-home message is I hope that both fields get together. I was yesterday on a discussion panel where it was said a lot that interdisciplinary research in your field is important and should be promoted. And I think that does also account to both fields of cellular and traditional agriculture. Thank you for your attention. I'm looking forward to questions and exchange. And also, sorry, thank you to ATA ATSAF, because they funded my congress this year. This is why I'm here. They asked me to hold this presentation, and I'm very happy that they asked me to do that. Thank you.
Thank you.
My name is not
okay, sorry. Sorry, thanks. Yeah, are there any direct questions to Robin? Questions of understanding or something, some additions? Yeah.
Pretty.
No, very simple monetary questions, because somehow the technology matches what consumer may expect, right? And other factor is also the price. By doing all this coloring of different colors and optical structure, how expensive or what is the trade-off, right? Because in one aspect, maybe consumer need this kind of optical view, and another aspect is also maybe also economically also, right? That consumer can buy it. So what is the
So in comparison to conventional meats, so the goal is to make it as cheap as conventional meat, so having it at least under $10 per kilogram. We are definitely not there, I guess. So the product in Singapore it's hard to estimate, but it costs like still at like $100 per kilogram, I would say. You pay like 30 euros in a restaurant for a small piece of cultured chicken, right? And it also turned out that it's not what they promised. So it was just like 90–70% plant mass and 3% cell mass instead of like having a higher cell mass. So yeah, it's difficult to estimate what are the costs at the moment, I would say. Yeah. Thank you.
Sorry, my question is kind of funny. You know, I'll kill for a steak. So I want to find out, you did this in the lab, and what's the texture? How's the feel in the mouth when one has to eat it?
Yeah.
Compared to like a real steak.
So it really depends on who's producing it. So in case of my prototype, yeah, we tried it actually, but it's so far away from a real steak, I would say. The reason is, in the end, my product is, for instance, very stiff. So you have like more like a solution for that to make it more
More tender.
Yeah, more tender, for instance, and also what really could increase like the real texture of meat, cultivated meat, is like to have a 3D culture. So normally cells are like grown like two-dimensionally, like just on the bottom of a flask or vessel or whatever. But we see more and more in the biomedical sector, but also for cultivated meat, that we need three-dimensional cultures. And if you get the cells to grow like it does in your body, then we get closer and closer to the mechanical properties. I saw some publications from other labs who were doing much better in cell biology and everything, who like compared the mechanical properties of their prototype, the conventional meat, and the mechanical properties were very similar. And they made like a study, like a sensory study, and I don't know the outcome really, but it was like 7 of 10. Yeah, in fruitfulness. So yeah, but we're still far away. I think that's a
One question back, then next.
Yeah, thank you for nice presentation. In the first slide, you said that that technology will reduce the greenhouse emission at 90%. Then in the last slide, you said that that technology is a high energy demand. Then I think if it is a high energy demand, then to produce that energy, if you go to the production site of that energy, there will be producing a lot of greenhouse gases emissions. How do you explain that? Thank you.
Get the question of the GHG. Yeah, so the SCAs, for instance, for the GHG emission or reduction, take into account that cultivated meat is produced using renewable energies. This is what I mean, that we have to look very critically at those studies, saying that can be reduced by 90% or 50 or whatever. Because yeah, they're saying we use solar panels or we use air power or whatever to make it, I think, more acceptable to policy and public. But if you do it like they say in the LCAs, it could be possible. And this energy intensive technology I showed as an example is at the moment the most dominant technology because it works and because there are no alternatives. It's very difficult to develop a technology that really also mimics this texture of real meat. But I think there must be technologies that are less energy intensive, like screen printing, for instance. Used for decades and decades in mass production. So it would be an option, but let's see. Thank you for your question.
Yeah, thank you very much. I think it's a very interesting area and very innovative. And but as you have already responded to some question here, like about the consumer, for example, preference, how it should be, and also the trust, etc. In between, also you said, for example, in Singapore, what is good approximately. So I guess, I mean, if you, I mean, I want to know how far are you in this sector? I mean, in terms of, you know, validation. Do you have now a kind of protocol because of standard? Because obviously that's what will allow you to define, I mean, how the product should go, you know, in terms of creating this meat type of meat. So if you don't have this, but I don't know, there may be some publication, because that's the first stage to do, to have a kind of protocol that is setting the stage, you know, globally or universally. So if you want to move in that direction, because you also it's depend who produce it. So I mean, so if you don't have those standards already from the beginning, so I think it may be some kind of you know, yeah, confusion also.
Very important.
Thank you.
Yeah, very important question. Thank you. So, for instance, for the products in Singapore, so Singapore was the first country that said we want to be like the hub for regulations of those products. So they set up the regulations, and every company can apply with their product, pass those regulations to get the operation. Yeah, so doesn't. So, and then the FDA, for instance, of America says, okay, we also say that the product is allowed in our country when it has the regulations, like this process of Singapore. And Switzerland, for example, also thinks about to be the hub in Europe. So they will also look at Singapore and say, okay, we adapt these regulations. I think that's the best way. So everybody looks at Singapore, and if everybody agrees and it's kind of approved so far, then you could adapt this to all other countries. I think that's the most reasonable way to go. But still, I mean, there's still some questions like genetic modified organisms and so on. There's still a lot of question marks, and that has to be answered before. But yeah, very important question.
Okay. And if there are no other questions, I would like to kind of come in a general discussion because thanks a lot, first of all. This session is about the SDGs and how innovations can kind of support the achievement of that. And we all know that by 2030 we are reaching, let's say, the deadline for the SDGs. We all know that it's going to be impossible, most likely to reach them. Still, I'd like to kind of provoke a little bit, asking you, be doing research on innovations and being kind of course, far away from market uptake and from real, like probably real implementation. I'm always wondering how we can kind of speed up, and also looking at the SDGs, that the way how they are organized, the way how they are supposed to be meant, they are meant to be thought integratedly. Like they are, it's not kind of okay to just focus on one SDG and leave out the others. And I'm always wondering, also in my own work, how do you, in your research, how do you take into consideration more than one particular target? Like in your case, it's probably the food sector. In another case, it's maybe agricultural production, but how do you see room for integrating this? You mentioned the interdisciplinarity as a thing, but is this actually happening? And I'm just wondering, because I think I know it for sure at our institute and our university, it's always a challenge because you're so much tied to your one particular objective, to one particular thing. How do you take everything into consideration? I think your example, Dipali, with integrating something, bundling more than one perspective is a very interesting one. I wonder how you in the room see it, or yeah. Is there anyone who is willing to comment on this? Or maybe, yeah. Okay, thank you. In my case, I think there's a problem with so much publications and so much information. Most of our research is focused on giving policy recommendations, and it's been flooded. There's just so much information. So with my limited understanding, let's say you have a university, there's a department, there's a working group, and each person in that working group is working on a particular project with particular objectives, and maybe some of them are kind of like intertwined in addressing the SDGs. So in my opinion, if I were a policymaker tomorrow, I would rather that they are kind of like condensed. It's just too much information. Even as a PhD student, I find it overwhelming reading everything and knowing everything. And it's like you're here, you're there, and you want to do. And in terms of implementation, also, it becomes very confusing and also overwhelming what and what not to implement. So maybe on our parts as researchers, we should, try to collaborate more and reach a common goal. Yeah, so that's my own, my own input. Too much information. Reactions.
Yeah, maybe I can add something to that. I had 2 more slides where I wanted to show how can the fields where both of us like cooperate. And I found in literature that, at the Good Food Institute, that there are the centers of excellence in America. I don't know if you know about it. Administrated by the USDA, so US Agriculture Department. And it's like it enables working forces of interdisciplinary teams that work together on one solution. And there's already a farm bill to also recognize, for instance, alternative proteins, just to say it here as an example. And I think that's the way, like to bring all the disciplines under one roof. And then you have the knowledge together, and not any individual must seek for the information, but you can just share it and bring it together. And I don't know if there's any like center of excellence here in Europe or Germany or like together with African states or whatever. But I think it's necessary, and maybe we should put more effort into that to like build such centers also here.
Yeah, and maybe just my personal view on what you mentioned. I think when we talk about collaborations, it's probably also not only the interdisciplinary type, but also the more transdisciplinary, where you say, okay, you're also talking to people who will be in the end uptaking your product or uptaking your solution. So I think this kind of stepping out of the whole pure research perspective is also looking at the SDGs and how pressing the deadlines are. I think we have to somehow bridge some of the conventional innovation life cycles, just be a bit, more crossing through, I would say. Any other comments or additions? Yeah. You were just...
Okay, I would like to say that achieving and going in the direction where we have to go. Because we as scientists, look at the work that he's doing on artificial meat, the new sensor technologies. There is a lot that has been happening in the past years. And I think we made tremendous progress in technology development, in targeting technologies and so on. What's lacking is the next dimension. And that's not us scientists, this is a policy arena. And I think it's nice to say, it's easy to say it's politics, it's the policies that are missing, and that's why we are not achieving it. But when I look at what has happened in the last 15 years in terms of progress in research, it's tremendous. It has never been as fast as been any time before. It's not our role to translate this into policies. We may be able, maybe we have to get better in formulating our research findings in a way they are digestible for politicians. But I think we have done quite a bit in the tasks that we were assigned to in the SDG. So we are not that bad.
So thank you again. So actually, for my considerations, the main topic theme was the technological innovations. And I also working with this part. But in case of review, especially of technology, just we are excluding the institutional part or governance part. That the first presenter said the social innovations. That means just collaboration, cooperations. Because if you just make a material technology or innovations in laboratory, but due to the lack of the process of cooperations or social innovations like soft technology, it will not adopted at root level. So this is the problem. And also the previous, I also work with this. So previous literature only including the technological innovation, but excluding the process. How we need to build a new process, new innovations to deliver this material technology or technology message. So we need to also think about the soft innovations like soft technological innovations. This is my message. Thank you very much.
Thanks. There's another one.
Thank you so much. I think, yeah, we understand that starting from basic sciences, this monodiscipline, you cannot replace it. And there has been this increase in the direction of transdisciplinary research. But still, some kind of research demands that people do things in their mono groups, in their laboratories. But I think that if we want to link to the SDGs, we need to change the culture. So to really say, for every project design, it should be in a way that still part of the project, there should be a chance for policy dialogue. Yeah, as a package, as an activity within the project, so that there is this part of the scientific work, and there's a chance that the outcome of the scientific work is still discussed with maybe people at the policy, maybe at the national level, up to the European level, so that at least what is the harvest from the project should have a chance to reach the ears of the policymakers. And equally, maybe as a university, to try that to be part of networks where some of these findings can be disseminated. For instance, the Agri-Natura, if you are a member of Agri-Natura, through the findings, you can disseminate to other networks or to other, you are part of settings where these kind of results are presented and the policymakers are there listening. And somehow this could be factored back into the programming process. But point really is that we get comfortable with doing our specific things and doing good work, which maybe ends at the narrow level. But it is good that within project, we should try to stratify so that still in those projects, some people who can have an influence, who are connected to the policies, can still have preview to the findings, even starting from the lower level to the upper level as much as possible. Thank you.
Thank you for that comment. I think what is quite interesting there is also that who are you addressing with this demand? Huh? So I think there is one side is the researchers themselves who kind of could question their role in terms of how far should they go and how is it their responsibility to bridge up to others outside of the university. And I think the other question is, the other side is probably also the funding organizations for all those, for that research, because I think that's, at least from our experience here, is also quite challenging if you would like to design a project like this and if you would like to open up, you also need funding for that. And I think, at least from the Austrian perspective, it's not that plausible to find funding for such type of open end research. No, I don't know how this is in other countries, but for us, I know for sure. Okay, thank you, everybody. Thanks for coming, and thanks for the presenters to really give us a very, very broad perspective there. Thanks also for the discussion. And yeah, I think if there's no other questions, then I would leave you out. And I think for those who have still questions, or I mean, there's maybe time also to just discuss between. Okay, thank you.